Hello friends, welcome back to my YouTube channel Nesting Padasale. Currently we are discussing the answers for this DBT BITP examination. Okay, so first question is Samir was investigating the impact of lactate dehydrogenase knockout on glycolytic pathway. What will be the net NADH production that he would expect in the conversion of glucose to pyruvate? There will be 2 NADH molecules will be produced from 2 NAD plus molecules. Okay, the correct answer is 2 NADH. Okay, so this is a part 2 video, friends. Already I had published part 1 video where I had discussed the answers for question number 1 to 52. Okay, you can watch that particular video. Next, which of the following statement regarding splicing of eukaryotic mRNA transcript is correct? Okay, in acquisition answer key, they just mention exons are spliced out and introns are retained in the mature transcript. But we all know introns will be spliced out and exons will be retained in the mature transcript. Okay, so if you ask me the correct answer for this question is option number A, which says the several reaction splicing process involve the hydrolysis of ATP. So it's the correct answer. If you take option B, here they just mentioned splicing takes place in cytosol, but splicing will take place in nucleolus. At the same time, in option E, they just mentioned snRNAs will be retained at the end of the mature mRNA transcript. This is a wrong statement. Okay, the correct answer for this question is option number A. So how many of you challenge this particular question? Please do tell me. Next, which one of the following is commonly used for performing REPD reaction? The correct answer for this question is random decamer primer, which is an oligonucleotide primer consisting of 10 different nucleotide bases. Okay, whereas if you take this random hexamer primer, it is generally employed for the cDNA synthesis. Next, the dog infected with rabies may contain negri bodies. Okay, so this question is repeated from DBT VET examination. Okay, next there was a question asked from COVID-19 vaccine. Which COVID-19 vaccine is based on mRNA, that is messenger RNA? The correct answer is Pfizer BioNTech vaccine is based on mRNA vaccine. It's an mRNA based vaccine for COVID-19. Whereas if you take Covaxin, it is an inactivated vaccine and it is an inactivated virus containing vaccine. Novavax is an adjuvant based vaccine platform employed for COVID-19 and this Covishield is a viral vector based vaccine. Next, which of the following is a false statement about S phase of cell cycle? So the correct answer is nucleolus duplicates during S phase. This is a wrong statement. Whereas histones will be synthesized in S phase, at the same time your centrioles and DNA will be duplicated in S phase. Okay, so nucleolus is a region where your transcription of your ribosomal RNA will take place and further processing will be taking place, and it is a place where your ribosome will be assembling for the translation process. Okay, so this is the correct statement. Okay. Next is an analytical kind of question where they are asked what would be the approximate increase in intracellular concentration of chloride ion. Okay, the correct answer for this question is 1 nanomolar. So please do tell me how many of you are attempted this particular question. Next is water is lost in liquid state in some plant through an opening in leaf called hydrothodes. These hydrothodes are, okay, so the correct answer for this question is they will be remain open always. There is no regulatory mechanism to close them or to open them. They will be remain open every time. So this hydrothodes is generally present in vascular plants. So it's just a plant organ which is responsible for releasing the droplets from the leaf surface or from the leaf margin or in the outer surface of leaf. Okay, this organ is responsible for the process called guttation. Next is again analytical kind of question. A particular restriction enzyme called A1 recognition and digests the DNA sequence of AAGC. So the recognition sequence for A1 enzyme is AAGC and the recognition sequence of A2 restriction enzyme is AAAAA. Okay, so they didn't mention the genome of an organism having 50% of GC content. It digested separately with A1 restriction enzyme and A2 restriction enzyme. Now they are asking which of the following statement is correct. So for this correct statement is option A. The digestion with A1 will leads to on average shorter length fragment than digestion with A2. Simple. If you check the restriction enzyme, the recognition site for restriction enzyme A1 is AAGC. So here GC is there. Whereas if you check with the restriction enzyme recognition site for restriction enzyme A2, it's have only A. Here they just mentioned 50% of the genome of organism having 50 GC content. So ultimately when you digest the organism which is having 50% of GC content with A1 means it will be leading to the production of short length fragments when you digest them with A2 enzyme. Okay, so the next thing is regarding to cellulose. So cellulose in food is consumed by human. So it is consumed by human. It will be serving as a dietary fiber only because humans don't have cellulase enzyme to digest the cellulose. Okay, so cellulose doesn't provide energy or it doesn't play an important role in long-term storage. In humans, we have glucose, which provides energy, and we have lactose for long-term energy source. Whereas the cellulose is taken from vegetables and from some other pulses or fruits that will be serving as a dietary fiber that helps in the process of digestion. Next, which of the following marine phototrophs can utilize the carbon dioxide as a carbon source? Okay, the correct answer is green sulfur bacteria, which has a capacity to utilize the carbon dioxide as a carbon source. There are other carbon sources that are highly available that some organism will be employing methanol, acetate, organic acid as carbon source. If you take this halobacteria, they will be employing amino acid as their carbon source. Okay, so it depends upon the nature of bacteria. Okay, so here they are asking the phototroph which will be utilizing the CO2 as carbon source. The correct answer is green sulfur bacteria. Next, who introduced the chromosomal theory of inheritance? It was introduced by Boveri and Sutton. So correct answer is Sutton. That's the correct answer. He was the one who determined a concept called genes are the unit of heredity, and these genes will be located in chromosome. So next is also an analytical kind of question. They had asked the problem sum. So I don't know how many of you had given a correct answer for this question. So the correct answer for this particular question is 6 into 10 to the power of 11. Okay. Next is Janaki wants to purify a protein with a pI value of 7.5 and a buffer with a pH value of 8.5. The buffer pH is 8.5 and the pI value of protein is 7.5. Which resin will be most approximate for purifying this protein by ion exchange chromatography? So one thing you should note about ion exchange chromatography, if pH of buffer is greater than that of pI, then the protein will be carrying negative charge. Okay, so the resin which you should use is anionic resin that you should be using, which will be carrying a positive charge. Okay, so there will be a binding between negatively charged protein and positively charged protein, that are positively charged ions that are present in your anionic resin. So here if you check, the pI value of protein is 7.5 and buffer pH is 8.5. So here the pH of buffer is greater than that of the pI of protein. So ultimately protein will be carrying a negative charge. So it will be attaching towards anionic exchanger which will be carrying a positive charge with them. Next, which is the name? What is the name of international agreement for giving international priority to patent? So it was given by PCT, that is Patent Cooperation Treaty. It will be giving the patent a right to protect their rights from internationally. Okay, you can able to protect the patent in international standards. Okay, the organism which is responsible is Patent Cooperation Treaty, which is an international agreement that was made. It was made to give international priority to patent. Okay. The next is a plasmid was digested with a single restriction enzyme called EcoR1, which will be recognizing. So this is a recognition sequence of EcoR1, that is GAATTC. And it cuts after G. The insert is blunted. Okay, so what should be a best strategy to clone the insert into the digested plasmid? Okay, the correct answer for this question is that if you digest, the plasmid should be treated with the clone fragment of DNA polymerase prior to ligation. So what will this particular enzyme, that is clone fragment of DNA polymerase, will be doing? So they have two enzyme activity. So the first enzyme, they have 5' through 3' DNA polymerase activity, and the next enzyme activities they will be having 3' to 5' exonuclease activity. Okay, so based on these two activities that they can be employed for treating the plasmid. Okay, so that's a best strategy to clone an insert, because insert is having a blunted. So they will be having a 5' to 3' DNA polymerase activity. So by using that particular property, they can able to fill a 5' overhand. So the insert will be converted to a sticky end. Then you can able to use that insert for the cloning experiment. Okay, so you can use the clone fragment of DNA polymerase 1. Next is in Arrhenius equation for temperature dependence of reaction rate, K is equal to, they had given the reaction. So if you know the Arrhenius equation itself, you can able to give a correct answer for this question. Here they are asking, what does this X signify? Okay, so X signifies the activation energy EA. So the correct answer for this question is activation energy. Next, CRISPR stands for. So CRISPR stands for clustered regularly interspaced short palindromic repeats. Next, which GM crop has been approved for farming by Government of India in 2022? The correct answer is genetically modified mustard has been approved by the Genetic Engineering Appraisal Committee. Last year in the October month, they had conducted the committee and the government has approved the GM mustard to be cultivated, to be entered into the environment. Okay, so now in India, many agriculture sectors and agriculture plantation, they are cultivating a GM mustard. Okay. Next is a simple question. So for this particular question, the correct answer is option B. So if you know this particular formula, like required concentration divided by stock concentration into required volume. So if you check with the DDTT, the working concentration is a required concentration. It is 2 divided by stock concentration is 100 multiplied by required volume is 100. Okay, so if you calculate 2 microliter is the correct answer. So remember this formula, required concentration or working concentration divided by stock concentration multiplied by required volume. That's a formula you need to employ. Next question is, what is the function of photosystem 2 in photosynthesis? Okay, the main function of photosystem 2 is that it is involved in splitting of your water molecules. Okay, next, which one of the following is a tiger reserve? So they are asking question, which of the following is a tiger reserve? If you take Gir, it is a national park where you can able to find lions in there. Okay, so you can able to find a huge population of lion in their natural habitats. But here the question is that which is called as a tiger reserve. So the correct answer for this particular question is Simlipal, which is located in Odisha. Okay, so that's a correct answer for this question. Next is again a specific activity question has been asked. The correct answer for this question is 1500 curie. So please do tell me if anyone has attempted this particular question or not. The correct answer is 1500 curie. Next, which of the following yeast strains is used for commercial production of riboflavin? Okay, so the yeast strain which is used for the commercial production of riboflavin include Erythrothecum asbai. Okay, so this is the yeast strain which is industrially it is used for the production of the riboflavin vitamin. Next is, which chemical is used to inhibit mold growth in bread? Okay, so the correct answer is sorbic acid, which is a preservative that is employed in various industrial sector. They will be employing sorbic acid as a preservative. Next, which one of the following technique is used for label-free measurement of protein-protein interaction? The correct answer is isothermal titration calorimetry, which is based on the amount of heat released or observed when there is any binding of biochemical molecules. So this particular technique is widely employed to determine in the binding or to study about the protein-protein interaction. Next is, which of the following is Bragg's equation? The correct answer is 2d sin theta is equal to n lambda. Next, a purified plasmid is fully digested with the enzyme that will be recognizes only one site on the plasmid and runs on an agarose gel. Identify the correct band pattern from the following. Okay, so correct answer for this question is option B, that is the undigested plasmid shows multiple band. How come? The undigested plasmid will be covalently closed in nature. So DNA in their form that can able to exist in 3 forms. They can be in relaxed nature, or else they can be in supercoiled form, or else they can be remain in circular form. Okay, so there are 3 forms of DNA is there. 3 forms. So if you take supercoil, they will be running fast in the gel, followed by the relaxed DNA and followed by the circular DNA. Okay, so that's the reason why this undigested plasmid will be showing multiple band. Whereas if you take the digested plasmid, it is digested, so the covalently closed circular molecule will be converted into a linear molecule. So that's the reason that linear molecule, when it runs on the agarose gel electrophoresis, it will be showing one single band. Next, which of the following is true about transfer DNA or tDNA? Okay, so integration of the tDNA can occur at multiple different sites, apparently in random site in a plant nuclear DNA. So the integration of tDNA can occur in a different site, at the same time the integration is random in nature. Next, during evolution, mitochondria is originated from. So mitochondria is originated from a endosymbiotic bacteria. So they are originated, both your mitochondria and chloroplast are originated from a endosymbiotic bacteria. Next is cyber-green based quantitative real-time PCR depends on which following property of cyber-green. So cyber-green fluorescence increases upon binding to the double-stranded DNA. So this particular cyber-green, they generally binds to the double-stranded DNA, and fluorescence generally increase when it's bind to a double-stranded DNA. So correct answer is the option number C. Next is also analytical kind of question. They had asked the molecular composition of the lysate, that is amino acid A, and two dipeptides B and C. The correct answer for this question, all the three amino acids and dipeptide, they will be having a molar composition of 1 millimolar. That's the correct answer. Next is, what is the lifespan of provisional patent applications in India? The lifespan of provisional patent application is 1 year, that is 12 months. Okay, so you can refer frequently asked question which will be available in the IPR website or patent website of India. From that particular PDF only, FAQ PDF only, many question has been asked. Okay, so if you are going to prepare for next year for some other examination, don't leave that particular PDFs that are available in the official platform of their website. Okay, so next question is with regarding to which of the following is not a type of intellectual property, right? If you take industrial design, trademark and geographical indices, the indication they are coming under the intellectual property right. Whereas radioactive material is not a type of intellectual property rights. Next, what is the full form of IPR? It is intellectual property rights. That's the full form of IPR. Next, the primary enzyme responsible for direct reversal of UV-induced thymine dimers. So the enzyme which is responsible is here photolyase. This enzyme is present only in lower organism. In human, this enzyme is not present. So the enzyme responsible is photolyase. It is responsible for the direct reversal of the UV-induced thymine dimer by breaking the cyclobutane ring of the pyrimidine dimer. Next, what will happen when a short day plant and a long day plant both kept in a favorable condition for flowering are exposed to a short flash of red light in the middle of dark period? So here you can able to see this is with regarding to short day plant. If here they are asking, they are exposing a short flash of red light in the middle of dark. So if you expose a light in the middle of dark period, then there will be no flowering. So short day plant will be remain vegetative, while long day plant will be flowering. Okay. The next question, which of the following cell type is a myeloid origin? So lymphoid lineage include T cell, B cell, NK cell, while this megakaryotes, erythrocyte, granulocytes and macrophages, they will be belonging to this myeloid origin. The correct answer for this question is mast cell. Next, the nucleic acid gives absorption of 260 nanometer. The function is primarily due to the presence of, so it is due to the presence of nucleotide bases which will be absorbing at this particular wavelength. Next, the crossing of F1 to any parent is known as, it is known as backcross. Next, if a genetic disease is transferred from a phenotypically normal but a carrier female to only some of the male progeny but none of the female progeny, what is the pattern of disease? It is a sex-linked recessive nature. Okay, the female is carrier, at the same time only she can able to pass the gene only to the male progeny. Okay, in that case it is a sex-linked recessive. The next is an analytical kind of question. In number series given over here, identify the value of x. The correct answer is 92 and this is the solution. So first you need to start with 1. 1 plus 1 square is equal to 1 plus 1 is equal to 2. Then you need to start with 2. 2 plus 2 to the power of 2, therefore 2 plus 4, which is 6. Next you need to start with 6. Again you need to start with 15. You need to start with 31, like that. So here 1 square, then 2 square, then 3 square, then 4 square, then 5 square, then 6 square. In that case, the x will be 92. So 56 plus 6 square, 56 plus 36 is equal to 92. Next, which of the following cellular structure is not involving bacterial adhesion? There is a difference between bacterial adhesion and bacterial movement. So, capsule, specialized surface receptor and fimbriae, they are involved in bacterial adhesion. Okay, whereas this flagella is responsible for bacterial movement and hemotaxis. Okay. Next is, which of the following statement about adjuvants is false? Okay, the correct answer for this question is that it creates covalent modification in epitope. So this is a false statement. Okay, sorry. Whereas the adjuvants, they will be enhancing the bioavailability of antigen. It will be helps in antigen presentation. At the same time, it also plays significant role in chemokine response. And this adjuvant, they will be increasing the immunogenicity of the antigen. Okay, so the correct answer for this question is they said adjuvants will be creating a covalent modification in epitope, which is a false statement. Next, which of the following microorganism produces dextrin? The correct answer is Leuconostoc mesenteroids is the organism which will be producing dextrin. This particular question you can be able to find in GATE Excel paper and GATE BT paper. Okay. Next is, which of the following non-covalent interaction have directional character? So correct answer is hydrophobic bond, which is a non-covalent bond that have a polar nature. Next, if a two plasmid having a same origin of replication are transformed in a E. coli, the replication of one plasmid is inhibited. So this is how come. See, if two plasmid they are having the same origin of replication, mean that in case there will be a formation of plasmid incompatibility. Okay, since they are having the same origin of replication, there will be a fight between both the plasmid. Okay, so that's the reason why few organisms like E. coli, they will be having a negative regulatory mechanism which will be taken over by the process of RNA antisense mechanism. So it will be blocking the replication of one plasmid, thereby allowing the replication of other plasmid. So they will be having a negative regulatory system called RNA antisense mechanism. So next is also a specific activity analytical kind of question. The correct answer for this question is 60 curie micromole. Okay, so I don't know how many of you had given the answer for the numerical answering type question. So please do tell me if you had given, if you want the solution for the numerical answering type question. Thank you friends, thanks for watching this video.